Hi, everyone. Thank you for attending December's Cafe Sci. I'm pleased to introduce to you Professor Bo Liu, who will be talking about the double life of STING, how an innate immune guardian fights pathogens and relieves cellular stresses. Bo Liu is a research assistant professor and cell biologist working on ciliary signaling and the innate immune cyclic GMP-AMP synthase, cGAS stimulator of interferon genes, aka STING pathway. He received his Bachelor's of Science in Biotechnology from Wuhan University of Technology in 2011 and his PhD in 2017 from the Institute of Hydrobiology at the Chinese Academy of Sciences, where he was studying the assembly of chlamydinous cilia. He then joined Gregory Pazor's lab at Program in Molecular Medicine / UMass Chan Medical School in Massachusetts, where he used CRISPR screening to identify E3 ligases that regulate cilia-mediated hedgehog signaling. After that, he joined the Aging Institute at the University of Pittsburgh, UPMC, to explore the non-canonical functions of the innate immune cGAS-STING pathway. Now I'll hand it over to you.
Okay, good evening, everyone. My name is Bo. I think you know me from the introduction of Noreen. Thank you, Noreen. I'm glad to share you with our latest research about the cGAS-STING pathway. Okay, the title of my talk today is The Double Knife of STING, how an innate immune guardian both fights pathogens and relieves the neural stresses. It's a fascinating title, but it's hard to digest. Let me explain it for you. We will talk about a protein. Its name is STING. Like STING, it's a protein. And in general, we say it has two functions. And the first one is to fight pathogens like virus, bacteria, we say germs. And the second one is relieves the neural stresses. Let's see how it has these two functions. When I first got the chance to give a talk here, and I'm glad, I opened my slides. It looks like this. And I prepared these slides for a talk in the Aging Institute for Research Progress Report. But then a few days later, they sent me an email saying, the audience are not scientists or researchers, but they are a group of people that really have great passion on science. They want to learn more about the progress. So I closed my slides. Why? Why do I close my slides? Do I give up? Of course not. If I gave up, I want to be here today. The answer is in this book. I have this book here today. And it's a story about Thomas and his friends. And they found some T-Rex bones and bring it to a museum. And this engine is Sandy. Sandy tried to assemble these bones into a real T-Rex. And every time this Sandy made a mistake, these engines will say, let's say what these engines say. Yeah, that's not right. If we assemble like this, that's not right. Or this, it's also not right. A slide like this for a talk here, it's not right. So, if I made a new one, especially for you, for a talk here. So in the in the rest of my talk, if I say something or that make you didn't understand or you feel confused, you just say that's not right. If I made it, you can say this. You can say, yeah, all right, okay? Okay, let's start. This time it's a real start. But start with what? I don't know. This is my first time to give a talk to the public. I also don't know how to start a talk like this. So I Googled it. Google says you may start like you can tell me a story about your study. So I searched how to start a story. I found that a good story is always start with what long ago. Yeah, I found there is a book. It's a collection of stories. Its title is long ago. So I think maybe every good story starts with long ago. I should also start my talk with long ago. Okay, let's start my talk with long ago. Long ago, like there's no humans on Earth, right? Long ago, there's apes, monkeys on Earth. And long ago, maybe there are dinosaurs, there are fish. But how long? I cannot always say long ago. It's about, let's say, 3 to 4 billion years ago. Right? There are gems on Earth. And there were 2 gems on Earth. Only 2. In my story, there are only 2. And one is Jack. The other is Rose. One day, Jack was infected by a virus. Here, it's a virus. Its name is Car. Yeah. Jack was infected and he was sad. Yeah, we can see here, Jack is crying. But there was no time for crying. Why? Because Jack needed to fight Car to protect himself and Rose. But with what? Jack is a gem. So we can see there's a trident. Can Jack fight Car with his trident? I don't think so. The trident is the weapon of this Aquaman. It's not the weapon of Jack. So we can say Jack may fight Car with his immune systems, right? He has immune systems. So let's say, what is immune? It's a new concept to you, or immune or immunity. I got some definition from Wikipedia. It's like immunity is the state of being insusceptible or resistant to a noxious agent or process, especially a pathogen or infectious disease. Immunity may occur naturally or be produced by prior exposure or immunization. Well, that's enough. I think at this moment you should say, that's not right. It makes you confused. Let me make it simple. In general, we can say immunity is about discrimination between self and non-self, right? Everything that is non-self, the immune system will fight against it. So let's go on with my story. It's Jack opened his toolbox. It's his favorite toolbox. It's the drills, hammer, wrench, or screwdriver. But can Jack fight Car with this toolbox? I don't think so. So let's close it. Jack opened his immune toolbox. He has an immune toolbox. There are so many immune systems. In this map, we can say this is the all detected immune systems that occur in microorganisms. This text are their names. It's just a nickname or abbreviations. And these numbers represent the abundance, like just a percentage or something. But it's hard to, Jack doesn't know them. He can't make a choice what to use. Like what this RM represent, what's ABI, what's cBAS, what is retron, what is cGAS. He didn't know, so he can't make a choice. Let's help Jack. help Jack to make the choice to fight against the car. So, to know these immune systems well, let's divide this system into two categories. Only two this time. It's to make it simple. One is adaptive immune systems, and the other is innate immune systems. Let me give you some example, like in the adaptive immune system, like CRISPR-Cas or CRISPR. And for the innate immune systems, I will also give you some examples, like abortive infection systems or restriction modification systems or chemical defense. There are more, but I want to list it here today. So we now know in the immune toolbox, we can divide these tools into two categories or two groups, right? But what these two categories mean? What is adaptive immune? What is innate immune? We still don't know. Jack also don't know. Let's help him. But to explain adaptive immune, I have to tell you a story first. It's a romantic story. I found that in both Chinese and Western culture, there are stories like a man one day saw a woman, a lady, at the crowd, and he never forgot that lady. He remembers her forever. I found a lyric like one glance was all it took. Right? Okay. What's that mean? Is there any relationship, like connection to the adaptive immune we were introduced today? Yes. For adaptive immune system, it's specific, which means the host, once it's infected by the pathogens, it will remember the pathogens forever. Like Jack was infected by car for one time. Then Jack remember car. And the second time or the third time, like every time, car infect Jack again, car will say, nice to see you again. If he say, nice to see you again, which means he knows car, right? I don't know if it's romantic, but okay, we now know adaptive immune. And I will give you an example of adaptive immune systems. It's a CRISPR-Cas. Nowadays, people are always saying gene therapy or genome editing. I think you may also hear that. In general, it's based on this CRISPR system. So what is CRISPR-Cas? Let's forget its full name at this moment. I will show you how it's gotten this full name. So again, this is Jack, and this is Car. Car is a virus. He infected Jack and inject its DNA into Jack. Then Jack grabbed some fragments from Car, right? He grabbed something and processed it. Then insert it into the genome of himself. That's like, let me give you an example. Let's say Jack is a bank and the Car is a robber. And the Car entered into Jack and try to get some money. Then Jack took a picture of Car and expressed wanted. And he inserted this into his genome. And with time going by, Jack have a lot of wanted in his genome. You can say it's a cluster of wanted, right? And they are spaced, interspaced, right? There are some space between these wanted. That's how the CRISPR-Cas system got its name. It's a clustered, regularly interspaced, shorter palindromic repeats. This describes the property of this wanted. Okay, that's how its CRISPR-Cas system works. Okay. Let's say if Jack choose this CRISPR-Cas system to fight against car in my story, what will happen? It's a Jack is no longer Jack anymore. Jack is Jack 0.2. It's enhanced. And then it's possible that this information can spread in the population. So let's say it's spread to Rose. And then Rose is not a Rose anymore. It's changed its family name. It's Rose Dawson. Yeah. Okay. Now we know enough about adaptive immune, right? And we have to know the other kind. It's innate immune. So what is innate immune? It's I still want to tell you another story. It's also romantic. It's a movie. It's a 50 First Dates. And this lady has some problem with her brain. I remember it's an accident, or I'm not sure. So every day she will forget everything she experienced the previous day. So this playboy has to do everything he can to make the lady fall in love with him in the morning. Right, that's why it got its name 50 First Dates. So is there any connection to innate immune? Let's say, for innate immune, its properties, it's unspecific. Every time car infect Jack, Jack doesn't know car. He just fight against the car. He will say, nice to meet you. Every time, yeah. I will also give you an example of innate immune. Here, there are 3 subgroups for the innate immune, and in the abortive infection systems, there is a kind of innate immune. Its name is cGAS. It's abortive means the host will die. But if the host will die, how it's immune? Because that's bacteria. They sacrifice themselves to save the population. That's how it works. For the cGAS system, and we can say here is car. Car infect Jack, and he inject its DNA into Jack. And this was detected by a protein cyclinase in Jack. And this cyclinase will produce a lot of cyclonic oligonucleotide. You just think it's a chemical. It's produce a lot of chemical. And this chemical will bind to the effect protein. This effect protein then kill Jack himself. Right. And in my story, if Jack chooses the cGAS system to fight against the car, but here, he will die. But he is a hero. He sacrificed himself to protect the population, especially Rose. Okay, we now know enough about adaptive and innate immune. And we know cGAS. And cGAS is in germs. And how about humans? It's fortunately this pathway is conserved. We say conserved because they are similar. And here, in the bacteria, this cGAS system, like they have this cyclinase, these chemicals, and this effect protein. Also, in the invertebrates or vertebrates like hare, fly, frog, human, they all have these components. They are similar. This is evolution. We say this is evolutionary concept. So, but in the human, it's called cGAS-STING, not cBAS anymore. It's cGAS-STING. But what is cGAS-STING? I think I will explain. It's with another story. You see, I also started with long ago. So long ago, it's this time it's not too long, not 3 to 4 billion years ago. It's 1912 or 1912. There was a man whose name was Titanic. Yeah, it's a strong man. It's the strongest man on Earth. One day, Titanic cut his finger when he was cooking. And because he's strong, he thinks it's nothing. He didn't take care of the cut. And he got infected by Jack and Rose. In this story, Jack and Rose are bad guys. They are gems. They infect Titanic. And then the cGAS-STING pathway is activated, right? Let's see how it's activated the cGAS-STING pathway. So this is gems. When they infect humans and their DNA exposed to human cells, and it was detected by a protein whose name was cGAS. It's this protein combined to DNA. And then, like the cBAS system, this cGAS can produce cGAMP. It's a chemical, just like salt or something. And this cGAMP is a messenger. It's alert the cell, say, oh, it's dangerous. Take some action. So this cGAMP bind to STING. We can say all the components is here. It's cGAS-STING pathway. But what's the function of STING? For STING, it's a multiple functional protein. But before we go ahead, I will give you some brief instruction to cell. So this is a human cell. It's for a non-biologist, it's hard to understand what's this. It's a cell, but if I say it's a house, I think you know it. This is a house. Inside the house, there are a lot of rooms. You have different rooms, like some for sleeping, some for cooking, some for studying, some for play. And also inside the cell, there are some room, like this, and this. Right? We call these rooms organelles. They have names. I will introduce some. So the first one, it's the nucleus. I choose this because it's so big, right? So what's the function of nucleus? It contains DNA. It has genomic information. It controls everything. It gives orders to the cell. So what do you think it's this neck? We say nucleus is the head of the cell. What do you think it's the head of a house? I would say it's wife. I'm not sure if you agree with me, but I say wife, it's the head of the house, right? Why? Because happy wife, happy life, right? Okay, I will introduce you other organelles. It's mitochondria. Its shape looks like a bread, right? But it's not a bread. But it do have some connection to bread. So the cells need to eat. They get some sugar or something. It was eaten by this, you can think. And by eating, they produce a lot of energies. So it makes the cell warm, you can think. So to warm a house, we need a heating system. Right? We say the mitochondria is the heating system of the cell. I will show you another organelle. It's a lysosome. It's also new to you. It's an acidic organelle, which means it's like very acidic. So what's the function of this organelle? You can say there are a lot of proteins inside. So its function is to digest trash. So inside a house, we live there and we produce a lot of trash, right? Some are recyclable, some are non-recyclable. But we do need something to, like trash bin or something, or recycle bin. This lysosome, they can digest these trashes to reuse this material, because you can't say, oh, it's trash, I exclude it out. Then it will be bad for the body. You need to recycling everything you can. Lysosome is keep the cells clean. Everything trash or bad, damage, they will go into lysosome for reuse. And we have recycle bins in the house. So I would say these lysosomes are the recycle bins of cell. And these lysosomes keeps the cell clean. We say we have other names like janitors or cleaners. But like now it's the holiday season, right? You give gifts to others, but you also receive a lot of gifts. And then you have a lot of paper box. Then I think you need more recycle bins, right? So when the cells, they produce more trashes, they need more recycle bins, which means you can increase the number of lysosomes, right, to relieve the stress. There are more lysosomes. And okay, but how to increase the number of lysosomes? There's a protein controls it. Its name is TFEB. If this protein is activated, it will make the cell to produce more lysosomes. And then I will show you where STING is. Here, STING localized in this structure. I want to tell you what this structure is today. So it's localized around the wife, right? I would say it's the husband. Okay, STING is the husband. What's the function of STING? Because it's an innate immune protein, I would say when cells get infected, I would say there's a fire in the house, right? If you say a fire, the wife will say, darling, save me, help, right? Then the husband, the STING, husband will take the extinguisher to put it out. But that's not enough. Extinguisher is only suitable for small fire, For a big one, you need to call 911, right? So this STING also call 911. He tries to put out the fire himself, and also he call 911. So if you call 911, what will happen? I think the street will be blocked by police car, fire truck, and ambulance, right? Also, when STING call 911, you will get an inflammation, which means if you bite by an insect, then this area will be red, itch, and then swell. This is the inflammation, which makes people unhappy, but it still helps you fight against the pathogens. So let's continue with our story. It's a STING, it's activated, then it's take out its extinguisher and also call 911. This is the first function of STING. And what's the second one? It's the second one, it's we recently found that STING can activate T-fab. So let's see how. This is Titanic. It's healthy, very strong, and there's no infection. And when there's no infection, STING is not activated. It's sparse. Right? And then T-fab is also not activated like this. This is a matched panel. And when Titanic get infected, the cGAS-STING pathway was activated. And then this STING tried to take action. It accumulates into these spots. And the T-fab also activated. You can see at first, it's not in the nucleus, right? And when it's activated, it's accumulated in the nucleus, this structure, the wife. And we know that T-fab is a master regulator of lysosome biogenesis, as I told you previously. So which means we can say the second function of STING is lysosome biogenesis, which means it promotes the biogenesis of lysosome. And as lysosomes are recycle bins of cells, so we can say the second function of STING is producing more recycle bins. So I will make a summary here. Say for the cGAS-STING pathway, for the STING protein, it has two functions. First one is extinguisher plus 911. And the second one is just making or producing more recycle bins to relieve stress. But what's that mean? It's, we know it right now. But is there any use for this? Yeah, of course. And we know that in infection, this cGAS-STING pathway will be activated. But it's also activated in cancer cells. We know cancer is not good. It grows very fast. And during this growth, sometimes some DNA were exposed and then detected by cGAS. And this pathway is activated. When it's activated, the immune system will try to fight against these cancer cells to make it shrink, kill these cancer cells. But we can say this STING also has this recycle bins function. If we block this pathway, right, to treat cancer, this cancer cells will die faster. Yeah, this is one potential use of our fighting. And in another potential use is during aging. It's when people are getting old. These cells are also unhealthy, and they are also DNA exposed. This cGAS-STING pathway is also activated. And when it's activated, it has two functions, right? And this gives you inflammation and also recycle bins function. And we can think when we are getting old, there are chronic inflammation, and which makes people really unhappy. But this pathway, it's makes the cell refresh. So what if we block this? If we, when people are getting old, use some medicine or gene therapy to block this pathway, and to boost this recycling function of STING. People will be healthy, right? This may make people enjoy their retired life, and also like may expand their lifespan. Maybe people will be like, you walk on the street, people are 120, right? So let me make a summary of my talk today. We are talking about a protein whose name was STING. And this protein, it's an innate immune protein. It's in the cGAS-STING pathway. And this protein has two functions. The first one is extinguish its husband and call 911. Try to fight against the pathogens. And the second function is to activate T-fab to boost lysosome biogenesis. We say recycling. This is the two functions. Okay, this is the end of my story. And I would acknowledge the organizer for this talk. I would also thank my colleagues in the Aging Institute, especially Torin and the mentor, Jay. And I would also acknowledge my friend, Sufen and Zhao Yue and my wife, because I practice my talk with them. Also, I thank my daughter. She's outside. And because I steal this book from her. Wait, one more minute. I have some post-credits thing for you. It's because I told you two stories, right? Two big stories. And in the last story, Titanic got infected, and he died of serious infection. Perhaps he should have known more about immunity. And in the first story, Jack, he chose the cGAS system to fight against car. And he sacrificed himself to protect the population and Rose. And finally, may God bless them. And Rose and her children lived happily ever after. Thank you.
All right, we're going to come around with a microphone if anybody has any questions and would like to start us off. I'll also be monitoring the Zoom chat if anybody wants to submit some questions there.
Not so much a question, but I just thank you for making this very clear. Looking at that original slide, yes, I can understand why you want to use that slide for the rest of us.
When you talked about blocking one of the pathways for, for fighting cancer, and then the other one for prolonging life, there's a trade-off there, isn't there? If you block one, you could be inhibiting the ability of the body to fight off something.
Do you mean if we can completely block one, doesn't affect
Right.
the other one?
Right.
Yeah, we can, because it's this protein is very long, and the tail, let's say the tail of STING, it's mediate the first function, and the rest of the protein, like they mediate the second function. So we can make this protein only express the, not the tail. There are ways to do that by gene therapy.
We have a question in the chat. There must be a delicate balance to reducing STING's first function. How can you maintain this balance?
Well, can you repeat it? How to make the balance between this function?
To reducing STING's first function.
Reducing.
There must be a delicate balance to reducing STING's first function. How can you maintain the balance?
Oh, to make the balance between the two functions. It's, I think in general, if we want to, like for cancer, it's, if we can make our chemicals to accumulate in cancer cells, we can block the second function, recyclability function, right? It's there are many cancer drugs, and they accumulate in cancer cells. For the second one, we suspect it's mainly happens in the brain if we have some gene therapy tools that can specifically block the first function, the extinguish 991 function in the brain, not the other area. Because if you block in your skin or something, it's not good because you will get infected. But in the brain, in general, like in neurodegenerative disease, there are gene therapy ways to make those you say we use some virus, artifact virus to make a change to our genes in brain cells, so that we can block the first function.
Thank you. There was a follow-up comment just to say nice stories. Now I know about STING and Titanic. There's another question in the chat. What is the potential time frame for this technique or capability to become widely available to help reduce many forms of cancer or enable longer lives?
I think it's at first people need to, I think with the publication of our work, soon people will try to test it in mouse, and then people can do clinical test. I think in 10 years, maybe people can use it.
There was another question. Are any of these technologies being used now?
Definitely not now, because we only have this finding recently. So previously, people only know the extinguisher 911 function. It's like thousands of papers, people are studying this because it's very strong. It's like when you get infected, those we say phenotype or some induce some change. They're very obvious, like thousands of fold change. But the second one, they are weak, but it's important.
Thank you. Thank you so much to Professor Liu for that really informative talk. I don't have a topic for next month's Café Sci yet, but it will be that first Monday in January, same usual time. And we hope to see you then. Thank you.
Thank you.